Hello, my name is Tiago Silva, and today I'm going to present the workshop for ATAC-seq analysis that Dr. BERMAN and I created. ATAC-seq is a technique used in molecular biology to assess genome-wide chromatin accessibility, and in 2017, BRYAN CORCES created an improvement on this protocol that was now able to interrogate frozen tissues. So as a follow-up of this work, BRYAN CORCES and Jeffrey GRANJA, as part of the Drs. Greenleaf and Dr. Chang lab, they profiled the chromatin accessibility of 23 human cancer types in 410 tumor samples from the TCGA, the Cancer Genome Atlas. They have almost 2 replicates for each sample, and this data is now available in GDC, the Genomics Data Common from NCI. So the main idea of this workshop was that we could understand students or users how to download and understand the ATAC-seq data that's available and how they could use that data for analysis. So pretty much we developed this workshop in this link here, in this RPub page, which is an HTML with R code, where we are going to download the esophageal adenocarcinoma data, ESAD, and esophageal squamous cell carcinoma, ESCC, and we're going to look for which are the regions that are open in SAD compared to ESCC, and then we're going to visualize it as a volcano plot and a heat map. So if you just click here, you're going to have a full workshop description with links to the article. You have a link for the GDC, where the Genomic Data Commons portal, where the data is available, which is this page. And then you're going to have pretty much a description of all the libraries you're going to need to install with R and all the code. And then we're going to have a step-by-step how to analyze, visualize this data. So the first step is to understand the ATAC-seq data. So before we start, all the data that we used in this workshop is available in a Google Drive folder. So this is a subset of the files that are in GDC. And the next step is understanding the data that's available in GDC. So the first thing we need to understand is that the data has been aligned to the Human Genome Assembly AG38. And then we have pretty much 2 types of files, which are, I think it's better describing the supplemental materials from the science paper. So I'm going through the supplemental materials right now. If you go to the science paper, you can retrieve that article. And you can retrieve this file. And on page 6, we start describing the data that we have. So what they did is they did a peak calling for the 796 ATAC-seq profiles. So as I said, we have 410 samples, but we have replicates there. And the first thing is that they choose to have fixed width peaks. The main reason is making the count-based and multifocus analysis less biased to large peaks. And when you have large data sets merging the peaks to obtain the union, if you don't make this fixed width, then you could have one very large peak just covering a huge region, and then you would not understand which would be independent peaks. Another thing they saw was that each sample had its own quality and read depth, and that somehow was affecting that peak call. So pretty much the data from each sample, it's going to be one peak with a fixed width of 501 base pairs. Okay, so like you get the summit of that peak and then just create it, extend 200 base pairs downstream and upstream of that region. And then the next step is they removed the overlapping peaks. So you get like one peak. Let's say you have two peaks overlapping and you keep the most significant one. Okay, then you go for the next one that is overlapping. You compare with which one's the most significant one, and then you keep it. After you stop it, having overlap, you go for the next peak. And you do that so you could like just keep the most significant ones. So this first data is what they said. It's a set of fixed width peaks for its sample, and they call it sample peak set. Okay. Now, if you want to compare that data with other samples, we had that problem, as we said. We had quality difference where we had like read depth difference, and then we had to do some kind of normalization. So what they did, they developed a way to normalize the peak significance scores across the samples and cancer types after they did the peak call. So for that, what they did is they got the max2 peak score, which is like minus log 10 p-value for its sample. And then they converted a score per million by dividing each individual peak by the score of all peaks score in the given sample divided by 1 million. So pretty much it says like if I have like one sample has more than peaks than the other one, we're going to do some kind of normalization there where we could like reduce that bias. Yeah, and the main reason to do this was simply because like the sample quality or the read depth increases, the number of peaks called and significance is going to increase. So we have to control for that. Okay, so by doing this normalization, now we are able to direct compare the peaks across donors, okay? Okay, so right now what we want to do is like getting all the patients from one cancer type and understand which are the cancer specific peak set. So the way they do, they got all the sample peaks sets. So imagine you have like all the esophageal samples, grab all of them, and then combine the peaks into a cumulative peak and trim for overlap using the same iterative procedure mentioned above. So pretty much like overlap all the peaks and then keep the most significant ones that are overlapping. And then they call the reproduce peak if at least 2 of the samples had that peak and with a score per million of 5 bigger than 5. So this is pretty much what it's going to be our cancer type specific peak set. Okay, so we're going to have like all the colon cancer specific set, all the breast cancer specific sets. And the last set of data you can obtain is pan-cancer peak set, which is cross-cancer comparison. And they had like to do a renormalization here simply because some of cancer types had more samples and more quality. And then you had like to take care so you don't have like a bias toward those that have more samples and this higher quality. So they renormalized those cancer type specific sets, and then they did again that iterative overlapping removal technique they described it before. Okay, so if you go to the DDC web page, you have like the supplemental data files from the article, and the data that we're going to use is actually here on the bottom. So we have the ATAC-seq counts matrix. We have like a README file. Let me just open here, where it's going to explain you what the data is. So as I said, we have the cancer type specific count matrix, and we have the pan-cancer peak set across all samples. And then give you pretty much a description here. We're going to open one of the files just to show. Okay. And then you have the normalized ATAC-seq counts for the pan-cancer peak. You have the in both RDS file or tax file. You have also the same file as a raw counts, so not normalized. And finally, you have for each cancer type specific the normalized counts. So you're going to have like the counts for only the breast cancer, only for the colon cancer, normalized and raw counts. Okay. Then you have the ATAC-seq pick calls, which is like a bad file. Here it's again the description. Okay. You can open. So this one is a small file. I'm just going to open the pan-cancer peak set here just to show. Okay, it's a little bit it's downloading, but I have the data here. Okay, so this is our pick call. You have, let me just close here. You have the start of the pick. The chromosome, the start, the end. You can see we have 501 base pairs, the fixed width here. You have the name of the pick, so this is a ASCII specific pick. Okay. Then we have the score, annotation, percentage of CG, and percent of AT on that region. So this is the ASCII specific. We have the pan-cancer. So what's the difference here is that right now we have chromosome, start, end, and then you have the name. So it's going to identify to you which is the tumor type that had like that specific pick. So this is a adenocarcinoma, if I'm not wrong, specific cancer, specific pick. Let me see, you might have like the other ones at the bottom. ACC here. And then you have like the score again and some annotations. Okay, it's reading, but I'm going to jump for the next files while it's doing that. Okay, so here we have like pretty much like a bad file, just saying where the peaks are and they score. And then we have a lookup table with the TCGA identifiers. So let's just open this. I'm going to show it in Excel. I think it might be better. So here we have like an identifier mapping between the Stanford UUID versus the TCGA. So the samples for the ATAC-seq, they have pretty much this PAN prefix, which is a mixture between the cancer type and the Stanford UID. And then one of the things that you need to do during the workshop is mapping those IDs to TCGA, so you could retrieve, for example, metadata available in TCGA. Okay, so this table is very important. No, we don't save it. Okay. And then what you have is the big files where you could use a genome browser to visualize where the pick is. Again, if you download those big files, the name is going to be based on the Stanford UID, and then you have to rename it. Okay, so the workshop that we're going to do is pretty much compare ESCC versus ESAD ATAC-seq using a t-test. Then we're going to plot the results as a volcano plot. We're going to define a cutoff for that difference. And then we're going to get the most significant ones that are different and visualize as a heat map. And then we're going to get one region and use a bigwig file to plot. Here, for plotting the bigwig files, we are using our software, but sometimes it's just easier to do a genome browser where you can just upload the bigwig file. Okay, so briefly, the first steps is download the data, which I'm going to cover later, but we're going to split the data, as I said, in squamous cell carcinoma and then adenocarcinoma. Okay, we're going to have our data that contains both subtypes, and then you break up into the squamous and the adeno, and then you do like a t-test, as I said. Then we're going to have like the p-value and the estimate difference between both peaks, both the peaks count. Okay, then you can do like a FDR adjustment for the p-values, hypothesis correction, and then you can plot the results as a heat map. So pretty much in the heat map you have our two results from the t-test, which is the log2 count difference, and on the y-axis we have the FDR values, and then we do like a cutoff. So here, for example, we define the cutoff to be 2 for the difference between the log2 counts, while the FDR, if I'm not wrong, is 0.05. And then you get this two set of regions, ones that are like more open in ESCC, and the other one that's more open in ESAD. And then you can plot those regions as a heat map. So again, you have the peaks we identify that are like here in the heat map. The reddish means it's more open than the blue, like lower levels of the ATAC-seq data. Again, we plotted this as a z-score. So for those that don't know what z-score is a normalization where pretty much you center the data in the mean, and then you can do that to visualize better the results. So you do that for each row. So you get like one region, you center on the mean. So mean now is 0, okay? And anything on the left is negative, and then anything on the right would be positive. So by doing that, you can reduce the noise in the visualization. So imagine like I have one region here that normally stands for really high levels if it was gene expression, okay? And ones that are like really low. When you do that in a heat map, you would not be able to visualize very well. So that's why we do z-score normalization for heat maps. But I can give more details later, more examples in the workshop. And finally, here is just one example of the peaks that have been plotted. So the first 4 one here are squamous, and the other ones are adeno. We can see there are a couple of peaks here that are only available in the adeno, and we cannot find those on the squamous. Yeah, this is the last plot we have for the workshop. Okay, now we're going through the code. This is the HTML file that has all the code to perform all the analysis we want. We have this table of contents, so we can just go there and click. So the first thing is we're going to use R for our analysis. Here is a code where I have all the libraries we're going to use. Y, like read R, we're going to read the files. GBylinks has a code for a volcano plot. You can do like heat map with the complex heat map and circularized, and the big wig files you can do with the Cario plot R. And then we have a code that you can simply run to install the package. So if you go to R and just run this, if you don't have a package installed, like I don't have this package, it's going to install for you. The other ones, it's checking if you already have it or not. And then we have a link to the data files that we're going to use in this workshop. So if you click here, you have the RMD with the R code that I'm going to execute. You have the HTML, which is the same as the RPubs. And then we have the data. So here in the data, we have the bigwig files for ASCA. We have the log2 counts for ASCA. And then we have the peak calls, the cancer-specific peak calls for ASCA, and the pan-cancer specific calls, okay? And then we have a file with the results for the t-test, just because we want to make it faster. It might take like a couple of minutes to run it. Okay, just another thing before we start. Although we use like bigwig plots, the karyotype plot are in R, sometimes it's just easier to use a GENOME browser. So here we have like the WashU GENOME browser, just to show how you can upload the bigwig files there. So if you go explore the new browser, you can select like the genome here you want, like human, and then you say, okay, I have a local track here, and then you go there and you select all your bigwig files, and then that's what I have. You go to chromosome 20, okay, to see like a given region. You can see like the y-axis here, they have different values. You can set, if you right click I think, or control click, you can set let's say 300 here, or yeah, you can like play with the height of the scale. You can select this, for example, and then you let's see if it's working. Yeah, I can set like a fixed width scale here. I don't know, let's say maximum is 100, and then you have like the same scale for each one. So you are not like comparing 10 to 100 on the peaks. And then you can color if you want. Let's change. Okay, so that's pretty much how you can do it. And then there's some ways to like download a PDF file or something that you can use in our publications. Just an example before we start the workshop. Okay, you're going to download the data, and then here I'm describing again the data types. You have like the sample specific peaks. We have the cancer type specific peaks, and we have the pan cancer specific peaks. The next step, we're going to explore a little bit the data. Okay, so I just installed the file. We're going to first read the ASCA specific peaks. So we read the file, so we have like chromosome, star, and end, the name of the peak, score, and annotations. Then you can take a look like how many peaks do I have, 127k. I can take a look on the range of the score, like it goes from 1.8 to 1000, almost 1500. Then we're going to read the pan cancer specific peaks. So this is a merge of all cancer type specific peaks. So if you come here, you can now know like, okay, this peak is from ACC, but you can run this code here. So pretty much I'm getting this name here, this column. I'm breaking up, removing the last part. Okay, so I'm just going to keep the cancer name, and then you're going to see like I have 20,000 K ACC peaks within the pain cancer one. And the score goes from 1 to 2,500. Okay, so this is pretty much our data. The next steps, what we're going to do is, okay, I had 127 peaks, right? And then, but only 27 are in the pain cancer. Okay, so the first question is just double check, like, are all my peaks like overlapping with the ATAC-seq? That's the first thing. So if we get the ASCA peaks and the pain cancer, and then like, okay, just what's the overlap? How many peaks of ASCA overlap with pain cancer? So we have here almost our 127K peaks, right? So just to recall what happened, you just like imagine I have two overlapping peaks, one from the pain cancer, that's like breast cancer, and one from ASCA, and the breast cancer has a higher score. So what happened is if they are overlapping and the breast cancer has a high score, we keep only the breast cancer, and that's why although it's overlapping here, that peak wasn't the one selected to be specific. So we're going to take a look on that peak, just an example. So okay, so here is one peak that it's in our so this peak, for example, is not within our pain cancer peak, so it was removed, and then like just to show why it was removed. Okay, yeah, the name here is different, but that's fine. We're going to take a look on another one. Yeah, I think that's okay. This should be EACC here. Okay, so this ACC is within okay, yeah, okay, that's fine. So okay, the first code here, we're going to subset the ATAC-seq pain cancers. We have this peak, and we're going to subset with the ASCA file. So this peak here in the chromosome 2 started like 11, 25, 41 from the ACC is overlapping with the ASCA one. Okay, so chromosome 2, 11, 25, 41, 6, 49. Okay, you can see there is an overlap between those two regions. What happened here is the ASCA has a score of 6, while the ATAC-seq has a score of 22. So like if you have those two scores that it's bigger on the ACC, we would keep this the ACC peak, not the ASCA. So that's what we had here. Of course, this data, just to recall, the data we are using here that I can specifically had like a renormalization, so it's not actually the real data that was used to do this comparison, but just to give an example why the region that was ASCA specific is not in the pain cancer specific peaks. And again, just to show, just going through the data again, our peak has a fixed width of 502, 501 base pairs, as they said. Yeah, so like all peaks, we have pretty much this information. Okay, so after we go through the data and explain a little bit what were our peak sets, we are going to use the raw count, the counts, the normalized counts, and try to identify regions that are open in the squamous versus the adeno esophageal cancer. Okay, for this step, what we're going to do is we're going to grab the normalized ATAC-seq search counts for, not for the pain cancer. We're going to get the the cancer type specific, this one, okay, which is a zip file. So if you go through this file, you're going to have this cancer specific counts, normalized counts. Yeah, there's one information that is important to say here. Not sure if I showed before. Let's see if I have here. Some of those files you have a specific command for uncompressing it. Let me just see if I have here. Zip. I think it's star. Yeah. Some of the files that are within GDC, they have like a weird directory structure, and then they give you how you can like ignore that directory structure with this command here, strip components 8. That's pretty much for that ATAC-seq big week. So okay, just going back to the normalized counts. Okay, so just to show again, how does the data look like? We have 38 columns, 126,000 picks. We just give a head here. Okay, that's pretty bad. Let's show the first 3 lines, first 7 columns. Okay. Okay, so we have like our chromosome, start, and the name of the pick, the score of that pick, and then the counts for each sample within that pick. Okay. Again, like here we have our samples in the columns, and we can see like we have T1 and T2. T1 means like one replicate, and then T2 means the second one, and then you have like the same prefix for both. Okay, so like for example, these two are the same data, same sample, just 2 replicates. Let me get just a little bit more. Okay, so these two are from the same 2 replicates here, then this should be 2 replicates, and etc. And also, as I said, this is not like TCGA barcodes, and then we have this code where just to show where we have the mapping between the Stanford UID versus the TCGA, and this code here is going to map the sample's name to the TCGA. Okay. Yeah, another thing is just to explain TCGA quickly. Let me grab TCGA barcode on Google. I think there's an image here. Yeah, this is. So briefly, like the barcode is going to identify the sample, the patient, and the experiment. So pretty much like start with TCGA, so it's a TCGA sample. Then we have the tissue source site where where the sample came from, then the participant, and then the sample. So 0,1 means it's a primary sample. Like imagine that then the same person had a recurrence. We're going to have like a 0,2. It could also happen that the person had like multiple cancers. Then there will be like some changes on this, but it would always be able to identify which is the patient and then which is the primary sample, tumor sample. If it's like a normal sample, there is also changes on this. So the first step here, we are like cutting off into the 16 initial characters because we want like until samples, like okay, which is my sample. Just going back here, that's what we're doing here. We create a new column with the sample, okay, instead of case ID. The main reason is because we're going to get clinical data and then we need the sample name. And then we're going to rename. So if you remove the first 5 columns, you just have like samples, okay? And then we do a match, okay? So first thing, grab this, remove the underscore here. We can see that this file we have a dash instead of underscore. So I say, okay, grab the column names, change underscore by dash, and match those to the BRYAN prefix, okay? So let's say, okay, get the first one here, change the underscore to dash, and then tell me which is the line, the row here that it's the same. So it's like, imagine it's the 6th line, just one example. Then what I do is, okay, if it's the 6th line, just grab me that case ID. Okay, that's pretty much what we're doing here. So let me just do it here, and then if we do again call names, now we have TCGA IDs, okay? You can see that since I didn't add replicates here, the 2 samples here are the same. The next step, I'm using TCGA bylinks with this column called data prepare to give me the sample information metadata, okay? So pretty much I give the barcodes, and then it's going to give me this table and say, I think it's easier to look like this, okay, like my barcode, my patient, it's a primary solid tumor, it's going to give me some other informations like stage of the tumor, days to follow up. Okay, and this is important if it's like squamous or it's adenocarcinoma, okay? A year of diagnosis, etc. All the clinical data that we have available pretty much. Okay, so I'm going to choose, as I said, the primary diagnosis one. I'm just going to remove the NOS here from the column, okay? Okay, so the first step is, okay, so, and let me just improve a little bit so I can recall exactly what we did here. Oh yeah. So here we actually added into the name a prefix saying if it's squamous or adenocarcinoma. Simply we added that column, okay, as I said. Okay, we just like did pretty much a matching. Like if it's squamous, just write ESCC. If it's adeno, just write ESAT. That's what this file is doing here. And then we're going to replace the names in the counts matrix. So we have a file with the prefix there. What I do in the next steps is creating a summarized experiment object, which is a bioconductor data structure. I think it's, let me just open here the image. So this is a way where you can merge metadata and data together in a better way for data analysis, okay? So we have like pretty much 3 matrices. Like the first one is going to be my counts data. Then I have information for the samples, another matrix, and I have another matrix with the feature information, it's like the ATAC-seq peaks. So that's what we're doing here. The first step is this input is genomic ranges. So just like, okay, just create a genomic ranges from my picks, okay, and we're going to give some metadata information for those, which are this coordinate. So just to show quickly how our GRANJAs is right now. Okay, so I have my pick here, okay, with the metadata I want, and then we have our sample information with all the metadata information, which is this one here. We just did a left join of the samples information versus the sample IDs. And then you create a summarize experiment object with those three matrices, three informations there, okay? Missing values not allowed in row names. Just 1 second, let me just rerun this. Oh, they pretty much might have updated the files, so I need to update, okay? Okay, so row name counts. It's going to be the same as row name ranges, okay? And now this should work. So it's not working. Okay, so my counts is as matrix. I'm going to update the HTML on the website. Oh my god, okay. Row names. Okay. Okay. Yeah, this is the problem. So row names. Let me just check something. It has to be the same one as the counts. Oh, great. Names. Okay, so this needs to be updated in the okay. Okay, let's do again this part and see if it's correct now. Okay, now it's here. I don't know why I messed this up, but and then here it is. Let's double check this is new. Names here. That's great. Okay. Okay, so then we have our summarize experiment object. Okay, we still have our data. You can see we can access like row ranges. It's going to give my pick information. Just, okay, and then code data is going to give me the metadata from the samples. So like code data here, raw data here, access this one, assays, access the middle one. And then the next thing we do, we just add a replicates to the names, okay? So replicate 1, replicate 2, etc. Okay, then what we're going to do is compare the ESCC versus ESAD. So just, you can use either the names here, just which ones are ESCC, or just grab the metadata from the summarize experiment, okay? So then we have the index of the, which ones are ESC, okay, just show it's correct. Names of ESCC samples should be all ESCC, and that's what we have. We have the same for ESAD. Okay, and then you just access the matrix. So just to show again, like we get the count matrix here, and then we compare, we simply do a t-test between all the ones we see and all the ones in the SAD. We're using BRYAN here, which is nice because you can add progress time. If you want to do in parallel, you can just like set parallel to true, and then you can do like do parallel reduce, do parallel reduce, and then set like I don't know, I want to set 2 cores, and then you can run this. When you run this, you're not going to have the time. But right now I'm just going to load the results here instead of running this because of time. Okay, so now just to show briefly what is the result. So we have like our peak, our raw p-value, our estimate difference, and then the FTR, and then you can visualize those results as a volcano plot. So here I have a code from TCGA that pretty much does, it's like a wrapper around ggplot2. And then you should have the results showing here. Okay, great. That's what I want to show. You can do the same with ggplot2. You can take a look on the code if you want to. Like, you can simply go here, copy the name of the functions without the parentheses, and then you have the code of a given function. Great. So we set the FTR cutoff to 0.01 and the estimate difference as 2. Then here you just do a table saying like, okay, how much do I have that the FTR is lower than cutoff, and the absolute estimate difference is higher than the cutoff, and I have like 4,067 peaks. Okay, the next step is we're going to use complex heat map to plot those. So we're going to call these 2 libraries. We have here a palette of colors for the ATAC-seq that is the same one used in the, I think in the article from the Cell science paper. Then I'm going to create the first thing is the annotation for the heat map. So pretty much the heat map you do like you plot the matrix and then you plot some annotations on the left or right or up. So here we're going to give the samples annotation. So just say like which is if it's as squamous or adeno, and what like if it's like replicate 1 or replicate 2. And then we select only the significant peaks in this line here. Okay. Then you set the scale. Okay, I want my minimum, my scale to go to the minimum to the maximum. You're just okay. And then I want to show the names for only 18 peaks. Here's just a trick, like you can add names for all the rows, but sometimes since you have like 4,000, you cannot read it. So you can select like, okay, I just want to show a couple ones. Sorry, you're just showing 2 here, 1 and 18, one of each example. So let's draw this. It might take a while to plot. Like if you want to see all the options here, you can take a look in the manual of the software. I'm not going to explain the results of each step here. Yeah, this doesn't look like correct. Let me check what happened. It's very likely. Yeah, let me just do actually the z-score one. Just want to see if that's going to improve the looking of the heat map, or if we have a problem in the cutoff. Yeah, okay. So it's pretty much like the z-score implementation here. So just to show the difference, here is we're normalizing each row to center within the mean, and here we are not. So imagine like this peak, we have like a Howard count. Let's say like the first one goes from 6 to 7, and the one at the bottom goes from 1 to 2. So this is going to be pretty much blue, and this is going to be pretty much yellowish, and you don't see the difference. But when you do z-scores and then you center, you can see better the difference between the two subtypes. Yeah, that's why we do a z-score normalization for visualization purpose. Because simply much the color of the heat map is going to be driven if you don't do the z-scores by the highest and lowest values. Okay. Okay. Also, we were doing here replicate separately, but you can merge them. So this is a function where we can get the row means by groups. That's what we do here. So the groups would be the samples. So for each sample, just get the mean of each group. So just show the matrix merged, and then you don't have like replicate 1 and 2, you just have 1 which has the mean of 2. So pretty much like, okay, give me the values, give me the groups, and then what we do is for each group get the mean and bind the results. That's pretty much what we're doing here. Yeah, since we have like replicates, we're just going to like get a subset of the one replicate, so we don't have like the double of the initial data. Metadata has to be the same size. And then we do again our heat map, but now with I'll run both at the same time, the z-score and then not the z-score. And then we can take a look on the results. So again, we don't have the z-score, so pretty much the changes are not visible. And then when you have the z-score, you can see a little bit better the difference between the data. Yeah, just one thing that I want to explain about the z-score. Yeah, the z-score is performed by this scale function in R, and it works over the columns. So you have to transpose it, then you scale, then you transpose it back. If you want a column z-score, it's going to show, like, instead of the difference between samples, the difference between regions for a given sample. Like, you could see which is the region that's like more important for that sample. You can simply remove the transposition here. Yeah, that's pretty much what I want to show. Just go I'm going through the last part of the codes here. We had some other useful codes. The first one is to get the cancer-specific pics. As I said, it's a zip file, right? And then you had to map the Stanford UIDs to TCGA ID, and that's pretty much a way you can do that for all the data. So if you run this, you should have a file for the breast cancer, a file for esophageal, a file for colon cancer, etc. Yeah, the last part is the ATAC-seq. I'm not going to show here the execution, just go through the file. This code is a little bit difficult, but like the samples are named as Stanford UIDs, and then you have to use that table mapping to change to UID to barcode. That's pretty much what this code is doing. So for each bigwig file, just extract the file UID and just map to the table and then rename, and that's it. Then what I have here is a software. So those bigwig files are quite big. You can download in the UCSC website. There are two executable files. One is called bigwig to wig and wig to bigwig, where you can specify a single chromosome. So the files I have prepared here, which are chromosome 20, that's how I did it. Just like get my bigwig, just grab my chromosome 20, and then convert back to bigwig. Main reason was just to have like small files for this example. And the next one is using this plot R to create a visualization in R, although I still believe there might be genome browsers sometimes are easier for visualization. And for controlling like the y-axis here, you can see they are not with the same values, right? Yeah, here we normalize the data using the same package, and we are showing a link between I actually don't recall which link is this. Yeah, using Elmer, which is a software that we developed. So it's pretty much like I have a CPG and a gene, and then that CPG is here, and then the region is here. I think it's pretty much, I might be wrong here. I'm not sure if like it's a promoter region or if it's a distal. But yeah, sorry, sorry. So here is the promoter region. We can see that the peak is bigger in the EAC, okay? So the region is more open. But like the enhancer region where the CPG is located, it's only open in EAC and it's not in ESCC. Yeah, so the enhancer here might be controlled in the gene expression to be more expressed in EAC than ESCC. That's what we want to show the last step. But feel free to send us an email if you have any doubts on the code or it's not working. And yeah, I think that's it. I'll keep information for contact in the YouTube channel, and then I can share the resources with you. Thanks so much.